students, this video is for Activity 6. Activity 6 is identification of normal blood cells. So the normal blood cells, we have mature RBC, we also have mature WBCs, which include your band neutrophil, segmented neutrophil, eosinophil, basophil, lymphocyte, and monocyte. Okay, of course, we need to include a thrombocyte or platelets. So for this activity, your output should be drawings of the mentioned blood cells, and you should use your color pens, your crayons, and some other drawing materials. So for that, you should scan your output and submit it online. So basically, I have here a short discussion for this particular activity. So before I start with the lecture discussion proper for activity 6, just a recap for your previous activity. Okay, so since you're done with activity 5, so I will assume that you have already done making a peripheral smear, and by having an ideal smear, okay, next to that you should stain your peripheral smears. But before you use your stained peripheral smears, you should check whether your peripheral smears are well stained. For you to be able to know that your peripheral smears are well stained, so you should take note of these characteristics. So macroscopically, it should have a color of pink to purple. So using your eyes, okay, looking at your stained peripheral smear, so the color of the stained smear should be pink to purple. Okay, and then microscopically, okay, it should have colors for different parts of different blood cells. So RBC should have a color of orange to salmon pink. WBC nuclei should have a color of purple to blue. Cytoplasm should have pink to tan. And granules should have lilac to violet. However, for eosinophil, the granules are orange in color. And for basophil, the granules are dark blue to black in color. So once you have checked that these are the colors of your blood cells, then you proceed to locating the zone of morphology. So in the picture, you have an example of the zone of morphology. It is where your blood cells are not overlapping with each other, and at the same time, of course, you should also, other than not overlapping with each other, they should not be too far from each other. Okay, so just enough to have a bit of space between different blood cells. Okay, so again, macroscopically, your zone of morphology is between your thin and thick smear. Definitely not in a thin smear, definitely not in the thick smear, but between. Okay, you should use your microscope to locate your zone of morphology before you identify the blood cells. So first, normal blood cell is your mature red blood cell. As we all know, our blood cells are generated and synthesized in the bone marrow. So once they are not yet mature, normally they should be located inside the bone marrow. So once this blood cell is already going to be matured, then they should come out from the bone marrow, okay, and become a mature red blood cell. So normally, the size of a mature red blood cell is 6 to 8 micrometer in diameter. The cytoplasm color is usually pink to slightly reddish, and towards the center, the color becomes paler or clearer. And take note, your mature red blood cell doesn't have a nucleus, okay, and it also doesn't have any granule or granules. So the shape of your red blood cell is biconcave. Okay, so when you find that there is a nucleated red blood cell, because it is really possible, then nucleated red blood cell is not a mature red blood cell. It is still an immature red blood cell. So this is a picture of your mature RBC. So paler or clearer towards the center, biconcave in shape, no granules, no nucleus or nuclei. And then we have band neutrophil. Okay, band neutrophil is actually the youngest cell in the series to appear normally in the peripheral blood. The other name for band neutrophil is staph cell or stab cell. The normal size of a band neutrophil is 9 to 15 micrometer in diameter. It is usually elongated or band-shaped nucleus or sometimes curved or sausage in shape. The chromatin pattern of a band neutrophil is coarse and clump, and its granules are called fine granules. Okay, they are located in the usually in the cytoplasm area. Okay, so as you can see, curved shape, sausage shape, granules in the cytoplasm area. Okay, so next we have your segmented neutrophil. This is actually one stage older than your band neutrophil. Okay, this is actually the most mature stage of the neutrophil series. Okay, so 9 to 15 micrometer in diameter is the size of a segmented neutrophil. The color of its granules or specific granules is pink to rose violet. And sometimes segmented neutrophil do have 2 to 5 lobes, but normally it is only 3 to 4. Okay, if you have 5 lobes or if you have seen 5 segments or lobes in a neutrophil, then that is already called a hypersegmented neutrophil. And then for the chromatin pattern, usually it is coarse and clumped. So this is an example of these two, okay, are examples of what? Segmented neutrophil. What kind of segmented neutrophil? Hypersegmented, because 1, 2, 3, 4, 5, it has 5 segments or lobes already. Okay. Next, we have eosinophil. Usually, the size of a eosinophil is 9 to 15 micrometer in diameter. The granules, the color of the granules for eosinophil is usually reddish to orange, and the nucleus, okay, usually has 2 lobes or it is called bilobe nucleus. For chromatin pattern, okay, the chromatin pattern is usually coarse and clump. Example of eosinophil and another example of eosinophil: bilobe nucleus and orange or pinkish or reddish granules in the cytoplasm. Next, we have basophil. Okay, usually the size of a basophil is 10 to 16 micrometer in diameter, and the granules, the color of the granules is usually dark purple to blue-black, and generally it is unsegmented or bilobe, and rarely it has 3 to 4 lobes. Okay, but sometimes it is really hard to count the number of lobes since the granules are very dark in color. So sometimes the granules will overlap the view of the nucleus. Next, we have small lymphocyte. A small lymphocyte has a size of 8 to 10 micrometer in diameter. The chromatin pattern or structure is usually round or diffusely lumpy to compact. Its cytoplasm area or edge is narrow, okay, compared to the large lymphocyte. So the large lymphocyte, its cytoplasm area is wider compared to the small lymphocyte. Okay, and the cytoplasm color is usually light or dark blue. Okay, however, okay, this is your small lymphocyte. As you can see, the area of the cytoplasm is narrow. The color of the cytoplasm is blue, light blue. For the large lymphocyte, the size of a large lymphocyte is usually 12 to 16 micrometer in diameter. The nucleus is usually oval or bulging, somewhat looser chromatin structure than the small lymphocyte. So, as I've said, large lymphocyte cytoplasm is the cytoplasm area is wider compared to the cytoplasm area of a small lymphocyte. So another difference is that the large lymphocyte cytoplasm color is lighter compared to the color of the cytoplasm area of a small lymphocyte. So this is a large lymphocyte, wider area of the cytoplasm, lighter color, and of course, the shape is bigger compared to the small lymphocyte. Next, we have the largest cell in the peripheral blood. Okay, monocyte, that is the monocyte. The color of the cytoplasm of a monocyte is blue-gray, and the cytoplasm of a monocyte is called ground glass cytoplasm due to its many fine azurophilic granules. Okay, so the nucleus of a monocyte is kidney-shaped and may show slight lobulation and may be folded, showing brain-like convolutions. Okay, so this is your monocyte. It is bigger compared to your large lymphocyte. In fact, this is the largest blood cell in the peripheral blood. But again, take note that the largest cell inside the bone marrow is not the monocyte. Rather, it is the megakaryocyte. Next is thrombocyte. Thrombocyte is also known as platelets. Okay, so the cytoplasm fragment, this is a cytoplasm fragment of a megakaryocyte. Okay, by saying that, okay, let me give you a quick background. So your thrombocyte, by the way, is just a fragment of the cytoplasm of that largest blood cell inside the bone marrow. So inside the bone marrow, again, there is a megakaryocyte, which is the largest cell inside the bone marrow. And of course, the megakaryocyte normally doesn't come out from the bone marrow. Instead, the cytoplasm become fragmented, and the fragments of the cytoplasm that comes out from the bone marrow, okay, are called thrombocytes or platelets. Okay, so the cytoplasm color of a thrombocyte or platelet is light blue to colorless, and when you use an electron microscope, you can really see the granules of a thrombocyte or of a platelet, and the color is usually scattered pink or red. Okay, so of course, when you use a usual microscope, you cannot really see the granules because the platelets are really small. Okay, but if you use an electron microscope, you can really see through the granules of the platelet inside. So to summarize everything, here is a picture of all the blood cells, normal blood cells. The erythrocyte, by the way, is the other term for RBC. Sometimes it is called rubricite. And then you also have neutrophil. We have discussed the segmented and the band neutrophil a while ago. And then we also have basophil. We also have the monocyte. We also have platelets or thrombocytes. And then we also have eosinophil, okay, which has what? Okay, distinctive orange color of granules. Okay, and then the lymphocyte, which has a small and a large lymphocyte. Okay, so there you have it, the lecture discussion of our activity 6. Thank you so much.